Purified DNA fragments and PCR amplifications from agar gel have a variety of applications, such as cloning, labeling, and DNA sequencing. Purified DNA can also be used for a subsequent PCR reaction, in vitro transcription, and restriction mapping.
The importance of this step is critical for the success of further experiments. In most cases, DNA will be derived from molecular biology techniques such as PCR, restriction enzyme digest, or electrophoresis, and thus will be mixed with enzyme salts and possibly other DNA fragments that may inhibit the following steps of the experiment. A simple solution to this problem is to purify DNA fragments of interest. This often involves agar gel electrophoresis to separate mixtures of DNA fragments, followed by extraction of DNA fragment of interest from the gel. There are different types of DNA purification methods, such as DNA recovery by electrolysis into a trap of salt or in a dialysis bag in a membrane using agarose enzyme, hot phenol, magnetic pearls, or DNA fragment purification with GTG agarose, also silica columns. All of these methods have some advantages and disadvantages that are shown in the following table.
There are many different kits for DNA gel extraction using silica columns. The kit used in this experiment is PureLink Quick Gel Extraction Kit from Invitrogen, which allows rapid and efficient purification of DNA fragments from agarose gels with different Ti or TBE percentage in less than 30 minutes. It also provides a recovery percentage up to 95% depending on the DNA molecular weight, that could range between 40 base pairs and 10 kilobases. The column has the capacity to bind and purify 15 micrograms of DNA. Another kit from Invitrogen is GeneJet Gel Extraction Kit. This extraction kit uses a membrane technology-based silica as a practical spin column. The entire procedure is more efficient than PureLink as it only lasts 15 minutes. It has a larger size of DNA fragments that can be purified between 25 base pairs and 20 kilobases with the same recovery percentage and a higher capacity to bind and purify DNA of 25 micrograms and can process up to 1 gram of agarose gel. Invitrogen also offers the Snap UV Free Gel Purification Kit that uses non-toxic reagents to rapidly purify intact DNA fragments from agarose gels. The key to isolating undamaged DNA is the crystal violet reagent, which allows the visualization of DNA in the gel under ambient light, eliminating the need for ethidium bromide and exposure to UV light. Nucleic acids up to 20 kilobases are efficiently purified from the gel using the convenient Snap resin, provided ready to use in a spin column format. The resin eliminates organic structures which generate hazardous wastes. In addition, there are no time-consuming ethanol precipitations. To purify DNA fragments which have a molecular weight less than 1 kilobase, column extraction kits are the best option. The extraction kits, like the ones mentioned before, ensure a recovery percentage up to 95% and have a high capacity of DNA binding. On the other hand, to purify high molecular DNA samples, the most useful method is by the agarose degradation with the agarase enzyme, because it is the most recommended for high molecular weight samples.
The PCR was obtained, the product was purified to an electrophoresis gel.
The darker line in the gel is a PCR desired product, which is 480 base pairs. This section of the gel was extracted and purified. Once purified and centrifugated, the total amount of DNA was of 0.2176 grams. In order to get a more precise quantification of the DNA, an spectrophotometric analysis of a 1 to 100 solution was performed, obtaining a 0.2572 micrograms per milliliter concentration. In the gel, there is several results that can be observed. The first product lines contain DNA from human amplification products. This amplification is 480 base pair. The last three lanes contain maize DNA samples. These samples are degraded. The first sample of the maize DNA shows 3 lines that can't be clearly read. The other 2 are so degraded.
DNA purification is very important because in most times, purified DNA is used for many molecular biology techniques, such as PCR. Also, contaminated DNA may alter the results of the techniques, thus affecting the procedure. To evaluate DNA purity by spectrophotometry, it is required to measure absorbance from 230 nanometers to 320 in order to detect other possible contaminants present in the DNA solution. The most common purity calculation is determining the ratio of the absorbance at 260 divided by the reading at 280. Good quality DNA will have an absorbance 260 ratio 280 of 1.7 to 2. A reading of 1.6 doesn't render the DNA unsuitable for any application, but lower ratios indicate more contaminants are present. Concentration and yield can be determined after gel electrophoresis is complete by comparing the sample DNA intensity to that of a DNA quantitation standard. Standards used for a quantitation should be labeled as such and be the same size as the sample DNA being analyzed. In order to visualize the DNA in the agar gel, staining with an intercalating dye such as a thin bromide or cyber green is required. DNA bonding dyes compare the unknown sample to a standard curve of DNA, but dynamic fragment and plasmid DNA will each require their own standard curves and cannot be used interchangeably. If the DNA sample has been diluted, you will need to account for the dilution factor when calculating final concentration.
There are several critical steps for correct purification of DNA from aqueous gels. The time of exposure to the UV light is essential. Identifying the PCR product in the transilluminator is critical because long exposure can nick the PCR template. Another critical step is the trimming of the gel. After visualizing the DNA in the transilluminator, the gel slice should be trimmed as much as possible, and the excess gel does not contain DNA should be removed from a high purification yield. The washing step is also important because extra wash with washing buffer and ethanol can help get rid of catastrophic salt residue of the membrane. It is critical to make sure that the membrane is dry after washing. The results obtained in the DNA concentration of the samples was a good one for a PCR result. The results obtained for the electrophoresis gels in lane 3 and 5 were the correct ones. The other results show degradation or no presence at all. This can be improved through several steps, like avoiding DNA degradation or improving the technique during electrophoresis gel's performance.